because we started from a new concept in medicine. And if you agree, we can spend 15 minutes just to summarize the basics of PRM and to warm up. Please, as always, stop me in every second, every time you need to ask a question, or since some of you friends are already expert, why not to share with all the other colleagues your experiences. Always remember, I will be very synthetic, because you are, this is a privatissimum, so you are already expert. But never forget to refer your clinical therapeutical approach to the triangle of the therapy. This is what we do every time facing a patient, but also establishing a design for a clinical trial. Remember that the basis is always the drainage and the detoxification of the patient. Especially have in your mind the crucial role played by that wonderful medication that is Guna matrix. Because since we release our low-dose cytokines through the blood capillary in the extracellular matrix, and these molecules have to reach the transmembrane receptors, if you have a toxic matrix that in terms of histology mean an extracellular matrix that is dehydrated, that is instead of in prevalent sol condition a smooth, clean matrix, in prevalent gel condition, you will not allow the passage of communicating molecules, cytokines, hormones, neuropeptides and growth factors through the extracellular matrix to reach the cell. So think in terms of product like matrix to boost every therapy, to enhance the activity of the signaling molecules. The yellow part of the triangle, that is the PNEI rebalancing. It is our heart of the therapy, the capability to act not only on the symptoms, but on the deep causes of every disease, that is the imbalance in the psycho-neuro-endocrine immune network. We are the only ones able to speak, to talk to the cells of this network. They only understand the language of signaling molecules. But signaling molecules for us are our medications. In this way, you recover the balance and you are acting on the cause and not only on the symptoms. We will focus today especially on the yellow and on the blue part of the therapy. How to use the messenger molecules, since they are the main actors of our medicine. Just to be very synthetic and easy, when you think in terms of psycho, neuro, endocrine immune network, think for each of these three main homeostatic control systems in terms of scale, balances, because they work that way. We have to visualize every system like a scale. The first scale we study today is the immune system. When you speak about immune system, you always know that you are going to do something wrong, because if you are too easy, it is too easy. If you are a little complex, it is not complex, it is complicated. So I will try not to be too easy and not to be complicated. Just to remember something about our immune system. First of all, I want to attract your attention on the crucial role played by the APCs, antigen presenting cells. We have several different types of APC, for instance, macrophages, Langerhans cells, but maybe the most important antigen-presenting cell is the dendritic cell, the prince of all the APC, which is the work done by the dendritic cells. They have to catch the antigen, to process the antigen, and to expose some part of the antigen on their surface. This way, they present the antigen to the CD4 cell, that in this case is still a naive CD4 cell. Naive means they don't know what to do when they will be adult. According to the type of antigen presented by the dendritic cell, the naive CD4 cell will differentiate itself in specific T helper subsets: Th1, Th2, Th17, Treg. So, summarize this part. It is just a little scholastic part, but it's important to be all of us on the same line. Our immune system cells are so many, and they are part of big families. I want you to focus on two families of immune cells: CD4 plus cells, CD8 plus cells. From CD4, we are able to differentiate several TH subsets, especially TH1. They are involved in cell-mediated immune response. TH2, they are involved in humoral immune response. Treg, they are involved in immune tolerance. They don't trust each other, and they control each other in order to maintain an immune homeostasis. If Th1 are physiologically expressed, they are involved in cell-mediated immune response. But if they are overexpressed, they are responsible of inflammatory diseases. Th2, if they are normally physiologically expressed, they provide a very good humoral immune response. If they are overexpressed, they are responsible of atopy, allergic diseases. Treg, if they are physiologically expressed, they control Th1 and Th2 not to be too much exuberant, otherwise inflammation or allergy, immune tolerance. If they are overexpressed, they are responsible of some forms of immune deficiency. For instance, in the most part of oncological patients, you can observe an upregulation of Treg. Since Treg are so high, they downregulate Th1 and Th2, and that patient is living an immune suppression. From CD4, please remember also another very important T helper subset, that is Th17. They are the main responsible of autoimmune diseases. All the autoimmune diseases. Crohn's and ulcerative colitis, psoriasis and vitiligo, Hashimoto, thyroiditis and rheumatoid arthritis, and so on. They have been discovered in 2006, Cook and Bettelli, and they isolated the first Th17 in the pancreas because they are overexpressed in type 1 diabetes, that is an autoimmune disease. So, with these glands, you have a picture of CD4 cells. From CD8 cells, we differentiate a lot of cells, but two populations are the most important ones. And they are T cytotoxic cells. Thanks to them, we defend ourselves from viral infections. You remember, T cytotoxic cells are responsible of the specific cytolysis of a cell infected by a virus. But T cytotoxic cells are also responsible to kill cancer cells. So, good guys. From CD8 plus, we differentiate also T suppressor. How much important are T suppressors? Every day, when we get up from our body, some million of cells decide to de-differentiate themselves in a wrong way, and they can become cancer cells. Million of cells. It's not a big quantity, some million of cells, because my body contains 40,000 billion cells. Why every day I don't develop a cancer? Thanks to these guys. Because T suppressors, as soon as they see that a cell is de-differentiating itself in a wrong way, boom, they attack it. All these cells are related each other. So, I leave this scheme on the flip chart because it's interesting sometimes to recall it. So, the dendritic cell presents the antigen to the naive. According to the type of antigen, the naive, after the blastization, can become Th1, Th2, Th17, producing typical interleukins of every different type of T helper lymphocytes. Treg, they are there controlling everything, taking down the exuberance of Th1 and Th2. We are able, with low-dose cytokines, to talk to Th2, Th1, Treg, telling them, hey guys, wake up if they are a little lazy. Or to say to them, take it easy if they are overexpressed. I mean, here, not here because we are not in Guna, we are at Ramada. Okay, here in Milan, at Guna laboratories, we did a discovery that is really interesting. And it is how the cells of our body talk each other. The language used by the cells, and it is the language of signaling molecules. And in our laboratories, we also discovered the volume used by the cells to communicate each other. And it is a very low volume. I mean, it's a very low concentration. So it's beautiful to have the possibility to whisper to the cells, to recover the physiology of that cell. Every disease is an expression of too much or too little activity of these cells. With the right words, we can recover the homeostasis. In terms of balance, this is a very easy